Okay, now we have here the cancer predisposing genes. So when speak about the cancer predisposing genes, so these are the genes here that would eventually develop you to become your cancer. So try to code here for the production of your cancer. So this is type of the genes which try to affect here the exogenous conversion of your precancerous here to become actively carcinogenic form. Another one, this gene here also try to affect the host impair the ability of the host here to repair for the damaged DNA. Third one, also try to alter the host here ability to recognize the presence of your foreign antigen in the form of your tumor antigen. And number four, try to affect here the apparatus that regulate the normal cell growth and differentiation. So if this is one is acted upon here by the carcinogens, this would develop here to become your cancer. Okay, then we have the term proto-oncogen. So this one try to regulate the normal cell growth and differentiation by the help of your growth factors and your transcription factors. So again, this one proto-oncogen. So this is your precancerous gene. Again, this one is acted upon by your carcinogen. It become here your oncogen. So oncogen is the cancer-inducing genes. So again, oncogen here target of your carcinogen is acted upon by your carcinogens and eventually develop you to become your cancer. Then we have here the family of your carcinogen or oncogens. So we have the first one, we have the growth factor, example for that, your cis oncogen. Another one, we have your epidermal growth factor receptor. Another one, in case of your CRC oncogen, belongs to your membrane-associated protein kinase group of your oncogen. Another one, we have your membrane-related guanine triphosphate protein binding receptor. In the case of your RAS oncogen, it also belongs to your cytoplasmic protein kinase. And we have also here your transcription regulator, which is usually found in the nucleus. In the case of your MYC oncogen. Okay, then we have here the different oncogenes and their disease associations. For example, the oncogen, we have here the AB1, so this is associated with your chronic myelogenous leukemia. We have also here the MYC related to your Burkitt lymphoma, and MYC that would be predisposed someone to develop here the neuroblastoma, epidermal growth factor, and human epidermal growth factor 2 associated with your mammary gland or mammary carcinoma or breast. And we've also here the RAS oncogen will be associated with different variety of tumors. Okay, normally we have here the body's defense for the elimination of your tumor. So normally your tumor antigen here is being expressed as a foreign antigen, and normally your immune system should recognize it as a foreign. And once recognized, it would eventually try to mount an immune response in order for that to eliminate the tumors. But the problem with that, once you have the tumor, your body could not eventually try to suppress that one because there will be several mechanisms here for your body not able to prevent the growth of your tumors. So your host immune response here fail in the eradication or the prevention of the growth of your tumors. And number four, so your body immune system would have the capacity to eradicate and to eliminate or to kill the tumor. But then along the way, there's be problem wherein your body could not recognize or could not even kill that one. Okay, then we have also here the different effector cells which are involved in the elimination of tumors in our body. The first one, we have your cytotoxic T lymphocytes. Basically, the cytotoxic T lymphocyte here try to help eliminate eliminate the tumor once the tumor has been in association with your MHC class 2. Now, next, we have here your natural killer. It could also kill your viral infected cells, tumor infected cells primarily if that one is being coded here with your antibody through your antibody-dependent cell-mediated cytotoxicity. Macrophages also help here in the elimination of your tumor by producing tumor necrosis factor. Another one, we have also antibody here also help in the elimination by production here of antibody against your tumor antigens that would help you then to allows it to be recognized by production of your tumor markers. Okay, then we have here several mechanisms why your tumor here could not be eliminated in our body. The first one, the tumor antigen is poorly immunogenic, so therefore your body could not recognize that one is a foreign or not. So your body don't know here if could not recognize there's already something wrong that is happening in your body. The second one, the absence of your necessary major histocompatibility complex, like for example, which is needed here by your C-cytotoxic cells, and therefore once your tumor is not associated with your major histocompatibility complex, your cytotoxic T lymphocyte will not be activated for the elimination of that. Another one, the patient is immunodeficient, so all the immune system of the patient here do not work in the elimination of your pathogens or your tumor, I mean. And lastly, we have here the immunoediting, so elimination phase, where your body or your tumor here has already mutated, okay, several times, wherein your body could no longer able to suppress that one, do not eliminate that one because your tumor is too aggressive already, because it has undergone already here some several cycle of mutation, and therefore you could not eliminate that one. Okay, the next one, we have your cancer staging. So when we speak about the cancer staging, you are dividing the cancer into different stages. So it's very important that you need to classify the cancer into different stage, because the treatment would most likely be highly dependent on the stage of the cancer. So because there are some stage of the cancer wherein the patient's no longer applicable your surgical operation, surgical excision of the tissues. When you need to classify that one. So stage 1 is your carcinoma in situ or just localized tumor. Stage 2 would be already the involvement, try to spread already here your blood vessels. Stage 3 with involvement your lymph nodes. In stage 4, it try to metastasize already here the nearby tissues. Okay, then the severity of your tumor is highly dependent on the tumor size. So the bigger it is, so the more severe it is. The type of tissues involved, the involvement of the regional lymph nodes, the small stage 3 na yan, and metastasis here would already your stage 4 or the advanced stage. Okay, then we have here the pathology of your tumors. You could also classify your tumors as well differentiated or you're poorly differentiated. When we speak about the poorly differentiated tumors, it's a type tumor we're in. It resembles here our fetal cells or young cells. This is more aggressive compared to your well differentiated. Well differentiated, most likely similar to the normal cells. Then we have also here the T and M classification. T stand for the tumor size, and for the lymph node involvement, and the M would be the involvement or it would be the invasion or try to metastasize the other tissues already. So you could classify that one based on the T and M classification. Okay, now we have here the tumor markers. So when we speak about the tumor markers, so this is the marker or the antigen protein that's being produced here by the tumor coming from your tumor cells. So we could use this one in order for us then to identify the presence of your tumors. So we could classify the tumor markers as virally induced or chemically induced. The virally induced much more immunogenic compared to your chemically induced. Okay, we have here the significance and the characteristics of a very good tumor markers. Number one, the tumor markers used here to identify the population having a disease and for those who don't have the disease. And second one is used to screen here for the presence of the cancer. Third one is used to monitor here the recurrence of the disease. Of course, here by comparing the result with the previous results. So if when speak about recur here, try to the condition here try to go back or return back. So we are expecting that the result here of the tumor markers try to still increases over the period of time. It did not decline, so I mean to say try to recur. For the fourth one, the tumor markers eventually reveal the presence of the disease or condition here on the stage wherein it's still susceptible for the treatment. So what's the use here of your detecting for the cancer if the disease condition is already in the end stage wherein you don't have anything to do here but just let the patient die. So very important here that the tumor markers is able to reveal the condition, and that condition is still on the susceptible stage here, susceptible still to your treatment like your chemotherapy or even your radiation. Number 5, so the tumor markers could also be present here in the different body fluids like your blood or even your serum, your urine or any other body fluids, and there should have here an available test that would allow you to identify and to measure that one. Through a different method, pwede siyang chemical, immunologic, or even molecular diagnostic tests. Okay, we have here a characteristic of an ideal tumor markers. First, the results should eventually reveal here the presence of a disease and should not include all those having a normal condition. The second one should correlate well with the stage and the severity of the infections. So I mean to say the higher the value here should eventually correlates with the more severe infection or severe conditions. That's how you're going to interpret supposedly. Third one, the result should be reproducible here that would allow you to have a comparison from the previous and to allow you to monitor for the treatment response of the patients. Then we have here the first tumor markers identified would be your Benson's protein, primarily for identification of your multiple myeloma. Okay, now we have here the categories of your tumor markers. We could classify that one as your TAA, tumor-specific antigen. We have tumor-associated antigen, carcinofetal. We have also here the spontaneous. TSA, or your tumor-specific antigen, is actually induced by the chemical reagents. So this is primarily uniquely associated here with each of your tumors. And this one is not found in normal cells, and the ability here to do not cross-react with another tumor. So it's very specific for each of the tumors. Next, we have here the tumor-associated antigen. This one is your virally induced. This is a chemical here found in the surface, produced by your virus itself. This one could not be found in the virus itself, but rather is being produced here by the host immune system in response to the infection caused here by the virus. And this one is also virus-specific. So I mean to say, regardless of which part of your body is being infected, if it's caused here by your Epstein-Barr, then try to cause your Burkitt lymphoma, so that will be manifestation. So regardless of where in your body part, so very specific lang siya for the virus, for the infection. Like Epstein-Barr, so try to cause your Burkitt lymphoma. Carcinofetal antigen that are related here to this is the type of antigens being produced here during the fetal life or your embryonic life. So then we have also spontaneous antigen. It's a type of tumor antigens. For in this one, it's no known exact mechanism here to try to cause the tumors. Okay, now we have here the classification of your tumor markers. So we could classify that one as your enzymes. Example for that, we have the prostate-specific antigens used for diagnosis of your prostate cancer. Oncofetal, we have your alpha-fetoproteins for your liver hepatocellular, carcinoembryonic for your colorectal cancer. Hormones, we have beta-human chorionic gonadotropin for your trophoblastic medullary, calcitonin for your thyroid. Also here your adrenocorticotropic hormone for small cell lung carcinoma. Okay, the following are mucins. We have the CA125, your CA19-9 for your ovarian cancer, CA115-3, and 27-29 for your breast cancer. Immunoglobulin in the form of your Benson's proteins used to diagnose here your multiple myeloma. We have also your genetic alteration, your HER2, human epidermal tissues here, 2 or the HER2. That's for your breast cancer. We have also here your other proteins like your human epididymis 4 for the ovarian cancer, TG for the thyroid cancer, and we have nuclear matrix protein 22. We have also here your bladder tumor-associated proteins. Another one, we have your complement factor H-related protein for your bladder cancer. Okay, then we have here the screening test for your cancer. So for the colon cancer, you could perform here your occult blood test for the presence of the occult blood that will signify here your colon cancers. Cervical carcinoma would also be performed here by your pap smear. You could also have your breasts identified here to your breast self-exam or even your mammography. Testicular cancer for your digital rectal exam. For the laboratory test, you could perform here your biopsy with the cross and tissue microscopic identification of your cellular tissue alteration. You could also determine here serological test with your tumor markers. Another one, you could also have perform here your molecular diagnostic tests. Okay, then we have here the molecular diagnostic test here that will help you to screen for the presence of your cancer for the tumor. So first, we have here the cytogenetic studies identifying here the defective na chromosomal defects. Okay, so most likely that one associated for that we have example for that we have here the karyotyping. Okay, then another one we have here your nucleic acid amplification for detection of specific defects in the genes. Okay, by either alteration mutation, so molecular with the nucleic acid sequence defects. Primarily, example for that, we have your polymerase chain reaction. Another one, we have also here your fluorescent in situ hybridization that will try to use the nucleic acid probes. We try to bind with a specific sequence in your target sequence that would specifically identify here the position of your defects.